Welcome to the UniProt Tutorial Series, brought to you by the Genomic Education team at the Jackson Laboratory. This short video will demonstrate how to find protein structure and function information within a UniProt entry. We will begin in the UniProtKB entry for human angiotensin converting enzyme, which is abbreviated ACE. Each protein entry contains information about protein structure, function, and expression. To learn more about the function of our protein of interest, we can navigate to the first section of the main entry page, titled Function. This section contains a summary describing the known functions of the protein. Throughout the description, there are links to references of scientific journal articles and reviews that provide evidence for the functional information. If you would like to learn more, you can click the linked text to be redirected to the relevant article. At the end of the text, there may be tags, such as number of publications or by similarity. These tags indicate that the functional information was derived from a number of scientific papers or based on sequence similarity to a protein in another organism. Clicking a tag provides more information about and links to the relevant sources. Below the initial summary description, there may be additional categories of functional information dependent on the type of protein in the entry and how much is known about the protein. Since ACE is a relatively well-studied enzyme, the entry includes information about catalytic activity, or the chemical reaction that ACE catalyzes, cofactors, or non-protein molecules that are required for an enzyme to be active, activity regulation, or molecules that regulate the enzyme's ability to catalyze reactions, and kinetics, or the speed at which the enzyme catalyzes reactions depending on its environmental conditions. Below all of that information, there is a subsection titled Features. UniProt entries contain information on several different types of protein features, including functional features, post-translational modification sites, and protein variants. Feature information can be accessed either within the relevant sections of the entry or through the feature viewer, which is linked at the top of the page. This tutorial will focus on the functional features displayed within the main UniProt entry. The Features subsection within the Function section displays information about protein features that are important for the protein's function. First, notice that the length of the protein is shown in gray. For human ACE, this diagram shows amino acid 1 through 1,306. Underneath the gray bar, colored shapes identify sections of the protein that are important for its function, also known as functional features. These features are also listed in the table below the gray bar. ACE is an enzyme, so its features include enzymatic active sites. Other features include binding sites, cleavage sites, and other regions of interest. To filter for a particular type of feature, such as the enzyme active site, we can use the Type drop-down menu. This will display all of the relevant amino acid positions for that particular type of feature within the protein. Thank you for listening. To learn more about UniProt and its features, continue to watch videos in this tutorial series. For other resources from JAX Genomic Education, find us on our website.